Okay, Bio30, so we're going to continue that discussion on action potentials that we started in the last video. And so here we've got our neuron, and we've got, think of this as a action shot, okay? This is a pause in time. So we've got the nerve impulse traveling, the action potential being propagated down the length of this neuron. And so it's currently at position 2, which means that position 1, if you remember our chart, just got repolarized. Position 2 is currently depolarized, and position 3 is still at rest. It's yet to be depolarized, so it's still polarized. So position 1 has just gotten back to negative 70 millivolts. Position 2 is currently at plus 40 millivolts, and the action potential is currently traveling at that position. And then at position 3, it is still at negative 70 millivolts, and the action potential hasn't reached it yet. So we're moving from left to right. Here's a picture. I'm kind of covering it, I think, for you here, but you've got 3 sort of cylinders here in the notes, and so these are meant to be the neurons, and same thing, right? You have the stimulus traveling. So wherever the stimulus currently is, because remember, it's physically moving down this wire, down the axon. So wherever it currently is, it's going to be positive on the inside, negative on the outside, and everything else is going to be at rest. But then as the potential moves, you're going to have that negative on the outside, positive on the inside move along the length of the neuron. And as it finishes up in one spot, so at position 1 here, the action potential has just gone through this, so it is repolarized. Number 3 is still polarized because the action potential hasn't gotten there yet. And at number 2, we are currently depolarized because that's where the action potential currently is. And in a second, point number 3 will become depolarized, so positive on the inside, negative on the outside, and position 2 will become repolarized, much the same as position 1 is, as it moves down the length of the axon. Here's another image here for you. So just watch this animation, right? I'll let it start over here. So at rest, and the nerve transmission comes in, depolarized, repolarized, depolarized, repolarized, depolarized, repolarized. Okay, and I'll show it again here. As everything after this is currently still polarized. It hasn't become depolarized yet, but it's about to as the action potential reaches it. So notice how as this animation changes, the inside becomes more positive and the outside becomes more negative as the action potential moves. But then as it finishes up in a previous spot, it has to return to the negative 70 millivolt resting membrane potential. So the whole reason why I was trying to explain potassium coming in, sodium coming out, is because we have to explain how this propagation is actually happening. It's not just positive and negatives, it's about the movement of sodiums and the movement of potassiums. So some drugs obviously lower resting membrane potential and they make it harder to generate an action potential. Alcohol affects a neurotransmitter called acetylcholine in the brain. So alcohol basically reduces the resting memory potential to way below negative 70 millivolts. This is why you get kind of dumb and in a stupor when you consume alcohol, because in order now for you to go from this really reduced resting memory potential, let's say it's like negative 90 as an example, you have to go from negative 90 to negative 55 to reach threshold. That's a significantly larger jump that you have to go through in order for a stimulus to actually cause a action potential than it would have if it was normally without the drug from negative 70 to negative 55. So this is why individuals who are drunk commonly like die of like hypothermia or freeze to death in the winter. If they're drunk and you're outside, you don't feel cold. Oh, I don't need a jacket. I'm fine. You're not fine. You think you're fine because it's taking so much for the stimulus, the cold temperatures, to affect you. Your nervous system literally doesn't realize it's cold because you're currently at a resting membrane potential of negative like 90, which is not normal. You have to go through this entire jump to get to that negative 55 threshold. Okay, so really important video here just to sort of finalize what the sodium potassium channels are doing and how that's causing the action potential, how repolarization and depolarization is occurring. I'm going to show you this animation. So watch the dots as they move. So number 1, resting state. So currently we are polarized. All of our orange sodium are inside because the king is always inside his castle, and there's far more potassium, or sorry, the potassium, the Ks, are inside. King is always inside his castle, and there's far more sodium, green dots, outside the membrane. So the outside is more positive compared to the inside. our resting membrane potential is negative 70 millivolts, and all the channels are closed. When we have a depolarization event occur, so an action potential is being propagated through this length, you now have the sodium channels open and sodium rushes into the neuron. So the sodium channels, the green dots, are going to rush inside of the neuron, boom, and this is going to make the inside of our neuron significantly more positive. So now we get close to that negative 40, but not quite. We're maybe around, I don't know, close to that positive 40. So maybe right now we're at like positive 20. We haven't quite gotten positive enough yet. So to get more positive, we are going to, oh, pardon me, I'm sorry, I misspoke there. So during the depolarization event, the sodium is going to rush in. We have reached that positive 40 millivolts. So we are positive enough now. The action potential has occurred. You are right there at that peak. As depolarization, this chart up in the top right corner, as it's gone up to positive 40, your sodium channels have opened and kept pumping in sodium, allowing sodium to move in along its concentration gradient to an area of lower concentration where there's more of them in here now, so that we can get to a positive 40 millivolt state. Then, now that the action potential has occurred, we want to bring everything back down to polarization. So we have to repolarize. So the sodium channels close because we don't need them to stay open. They've already done their job. And now the potassium channels are going to open. Now potassium channels are going to move along its concentration gradient from an area of high concentration inside to an area of low concentration outside. Potassium is going to leave the cell, the neurons. So boom, there you go. Your orange potassiums have left, most of them. So now this has restored the negative charge inside of the cell. And now we have to get back to that resting state. So the sodium channels are closed. The potassium channels are closed. Now there's mostly sodium inside and mostly potassium outside, and we don't really love this. And so now we have to use a sodium-potassium pump to put the sodium back outside and the potassium back inside, where it can now prepare for the next action potential to occur. So this happens through active transport at this sodium-potassium pump, where it brings 3 sodium out for every 2 sodium that come back in. And it'll keep doing that in order to bring us back to the negative 70 millivolts. Okay, synaptic activities. So how does the information get from one neuron to another? Well, neurons don't actually touch. They just sort of straddle each other very closely. And you need to have that information, the electrical signal from one neuron, somehow jump to another neuron so more action potentials can occur. And the only way that's going to happen is if you have that communication. So the communication is going to occur in this space called the synapse. And the synapse is this precise small gap between 2 neurons. The dendrites of a receiving neuron and the axon projections of the end of a previous neuron. So transmission across the synapse is slow, but there's lots of synapses. So synapse is that space between 2 neurons. So we've got pre and postsynaptic neurons. And so an impulse moves from the presynaptic neuron to the postsynaptic neuron. So from the neuron giving the information to the neuron receiving the information. The nerve endings of the presynaptic neuron are filled with synaptic vesicles. So a vesicle is a chemical sac or a cellular sac which contains chemical messengers, which are called neurotransmitters. So remember, hormones are the communication messengers of the endocrine system, and neurotransmitters are the chemical messengers of the nervous system. So the pre and postsynaptic neurons are here. We call it a presynaptic neuron because it's the one giving the information, and we call it the postsynaptic neuron because it's the one receiving the information, and the synapse is the space between, where the signals or the neurotransmitters are going to have to get sent across. Think of this as a street. You're crossing a street, you're on one side, the street that all the cars are passing is the synapse, and the other side of the street is where you're trying to go. That's the postsynaptic neuron, and you're currently standing at the presynaptic neuron waiting to cross. So there you go again. There's the postsynaptic neuron, there's the presynaptic neuron, there's the synapse. So you're over here, you are trying to cross the street, and you're trying to get over here. So vesicles release neurotransmitters. The vesicle comes up to the membrane of the presynaptic neuron and just splooges it out. All the neurotransmitters are in the synaptic cleft, and then those neurotransmitters are going to bind to receptors, very specific receptors, on the postsynaptic neuron, which will create some sort of a signal, either an excitatory signal or an inhibitory signal, a turn off or a turn on signal on the postsynaptic neuron. Okay, so I really like this picture. Let's just kind of discuss what's happening here. So remember, if we start at step number one, the neurotransmitter is synthesized in the cell body, it's transported down the axon, and it kind of hangs out in vesicles near the presynaptic portion. So now I'm going to zoom in over here. So there's our vesicles. When that information is propagated in those polarization, depolarization, repolarization, action potential events that we just went through on the graph on the previous video and earlier in this video, gets propagated all the way down to the end of this axon, it's now going to tell these vesicles, which carry very specific neurotransmitters, whichever neurotransmitter is involved in that particular part of the body and that particular neuron, is now going to bind to the surface of the presynaptic membrane, and it's going to splooge, very scientific word there, splooge or release the neurotransmitters into the synaptic space, also called the synaptic cleft, which is the gap, the street that you're trying to cross. And it is going to involve calcium ions to help release those neurotransmitters. Once those neurotransmitters have reached the receptors on the postsynaptic neuron is going to have some sort of an effect based off of what the neurotransmitter told them to do. And then the neurotransmitter is going to get recycled and taken back up so that the presynaptic neuron can take the leftover neurotransmitters and reuse them for the next time it has to release them. So we are going to use acetylcholine as an example. It's a neurotransmitter, it's a common one, but it's not the only one. And so I'm just going to show you this diagram here. So the impulse arrives at the presynaptic neuron. Our acetylcholine, our yellow triangles are inside of these vesicles. Calcium is involved in the release of the neurotransmitter from the vesicle. So once it has kind of entered, it's going to stimulate the release of the vesicle's contents. So the neurotransmitter, in this case acetylcholine, is going to get pushed out or splooged into this synaptic cleft or synaptic space. The yellow triangles, which are acetylcholine, are now going to bind to their receptors on the postsynaptic neuron. So there they go, binding to their receptors. And then the sodium is going to rush in on the postsynaptic neuron, causing a depolarization event on this postsynaptic neuron, and then boom, you got the same action potential that originally happened in this neuron, now happening in this neuron. But they never actually touched. So it's as if you drove your car. Same example as a couple of videos ago, right? You're driving your car, and every time you drive your car, you create holes in the ground, so somebody has to come behind you and fill those holes before the next car can drive by. That's basically the action potential, the depolarization and repolarization events. And now imagine if at the end of the road there was water and a river, and you had to cross that river. The only way you can cross the river is if you got out of your car and hopped in a boat and then floated across the river and then got to the other side where you got into another car and now you started driving. And as you drove down that road, same as before, you caused this depolarization, repolarization event over and over and over again where you drove your car, you made holes when you drove your car, so behind you was a pothole filler that allowed another signal to come behind you, another car to come behind you. And this might happen over and over and over again where you keep having neurons, neurotransmitters transmit that information over and over and over again until you get to your destination. There's no such thing as a long neuron that's going to go right from your brain to your big toe. It's going to have a series of neurons, and throughout you're going to have these synapses connecting them. Okay, so now you have your depolarization event, and boom. Now, one last thing, cholinesterase is an enzyme. Anything ends with ASE is an enzyme, and it's going to break down the acetylcholine neurotransmitter and allow it to be reabsorbed and reused by the presynaptic neuron the next time it needs to transmit that same neurotransmitter. So there you go, it gets reuptaken and it gets reused. So the nerve impulse arrives at the presynaptic membrane. Calcium ions and sodium ions are going to rush into the presynaptic membrane and allow for the neurotransmitter to get released from the vesicle. the neurotransmitter, acetylcholine in this case, is going to attach to the receptors on the postsynaptic membrane, and that's going to cause the sodium channels to open and a depolarization event to occur and an action potential to happen in the postsynaptic neuron where that signal can now get propagated. An enzyme, in this case cholinesterase, is going to break down the neurotransmitter acetylcholine and reuse it. And so then the presynaptic neuron can take it back up and use it around in the next round. So that is pretty well the discussion I would like to have with you on action potentials and neurotransmitters. So in the next videos, we're going to talk more specifically about specific body systems that the nervous system interacts with. Thanks, guys.